Without further ado, I'll hand over to the next speaker. This is Ognian Neytchev. Paul Shields was originally scheduled to speak, but Paul couldn't make it. But Ognian is a colleague of Paul's, so we're going to hear the story directly from Ognian. So I'm really excited for this one. So Ognian is at the University of Glasgow, and he's going to talk about using epigenetic clocks to track age-related physiological function in the kidney. Over to you, Ognian.
Thank you, Peter, for the introduction, and to all the organizers for having me. And thank you, of course, to all the participants for joining, especially those who are joining from not the most convenient time zones. Yes, I'm Ognian. I'm a member of the Shields Lab at the University of Glasgow. Our topic of research is mainly aging and the roles of various socioeconomic factors and biological processes. As we probably all know, biological aging is an increasing problem as a result of the aging of the global population. This is true both in developed as well as in developing countries. The number of old people is increasing everywhere, and it's predicted to keep increasing in future decades. On one hand, this is a good sign because it means that lifespan is increasing and people are living longer. On the other hand, it is not so good because health span, or the number of years that people are able to enjoy good health, is not increasing by an equal amount. So this causes an increasing burden of morbidity and multimorbidity in the population. This has a cost for society because these elderly people who have chronic conditions require help and other forms of care. And it's also a burden for the individuals because it reduces their quality of life. It limits their ability to enjoy their life and live as they would like to. Biological age comes with a constellation of different diseases that are sometimes referred to as the disease of aging. They're all different diseases with different etiologies, but they share common underpinning mechanisms. Some of these are quite well known in the general population: cancer, cardiovascular disease, neurodegenerative diseases. Some are perhaps somewhat less known, but are still quite important. For example, chronic kidney disease, or CKD, which will be one of the focuses of this talk. So CKD is a disease that is increasing in prevalence. It has reached epidemic proportion in many countries and is projected to keep increasing with the aging of the population in future decades. The scientific community has identified a number of common hallmarks of aging that are quite well conserved across different taxa, all the way from flies and worms to humans. The original paper from 2013 from Lopez-Otin and other co-authors was perhaps one of the most cited papers in this field. It was just updated a few weeks ago with the addition of 3 novel hallmarks that in the recent years have gained increasing attention as they play an important role in the biology of aging. These are disabled macroautophagy, microbial dysbiosis, especially in the gut, and chronic inflammation. Among the original hallmarks, I would like to highlight a few that will be particularly relevant for this talk. Telomere attrition. This is the shortening of the ends of linear chromosomes that happens with cell division, as well as a cause of other factors such as oxidative stress. Another one is epigenetic alteration, in particular DNA methylation, which is very important for expression of and transcription. It regulates both the transcription of protein coding genes as well as other non-coding RNAs, including transposons that are all dependent on this genome-wide DNA methylation pattern. Finally, I would like to highlight cellular senescence. This is a permanent exit from the cell cycle that cells undergo typically due to some kind of molecular damage that cannot be repaired. Senescent cells tend to accumulate with age, and they are thought to be, over the long term detrimental for the local tissue because they can disrupt the local environment. They can precipitate senescence in neighboring cells, and they can also cause inflammation by secreting pro-inflammatory factors. In spite of this significant progress, there are still a number of questions that remain at least partially unanswered in aging research. First of all, we still don't really quite understand how do we age, what is the exact mechanism sequence of events that leads a young person to become old biologically. We also don't fully understand the role of the environment, lifestyle, and deprivation into precipitating and accelerating aging. And also, we have a methodological limitation, which is the absence of a clearly established and universally validated measure of biological age. There's still debate about how to define biological age and how to measure it, and which is the best way. An important concept that we think is key in understanding biological aging is antagonistic pleiotropy. This is the idea that a certain trait or gene can have multiple effects, and they can be antagonistic. So some can be positive, others can be detrimental. This is particularly relevant when the positive effects are expressed early in life and the negative effects are expressed later on. Because when we have this situation, evolutionary pressure can select this particular trait because it gives early in life an advantage in terms of perhaps survival or reproductive success, even if it comes at the cost of greater aging and frailty later in life. Another crucial factor is the role of the exposome. This is a set of all exposures that an organism is subjected to. This includes everything from chemicals to nutrition, micronutrients, as well as socioeconomic factors such as social environment, personal relations, psychological stress, etc. The exposome has a very important role. In fact, there are 3 exposome factors: air pollution, tobacco smoke, and diet, that collectively are responsible for about 50% of global mortality. So we believe that this is a very promising lever for intervention to increase health span, improve health at later age. As I mentioned, our lab is based in Glasgow, and if Scotland is the sick man of Europe, Glasgow is the sick man of Scotland. When we look at mortality rates in different areas, we see that Scotland is at the upper end of the distribution within Europe. But then when we look closer, we see that most council areas are actually comparable to the rest of Europe. However, there are in particular these 3 outliers that are all deprived areas around Glasgow that have a much higher mortality rate compared to the other regions. This creates a very stark socioeconomic gradient. For example, if we compare Colton and Lenzie, which are 2 council areas around Glasgow that are only 12 kilometers apart from each other, they have almost 30 years difference in male life expectancy. Also, when we look at the map, we can see that affluent and deprived areas are very close to each other, but still they have this very strong difference in outcomes. It is also important to highlight the fact that deprivation is not only a matter of absence of income and money, but it is also an absence of other factors such as support, and other resources and opportunities. So it's not a simple thing to solve with just giving more money to that community. Another important aspect that we think is useful for explaining this gap between affluent and deprived areas is the concept of allostasis. This is a pressure that is resulting from either physical or social stress on the body's ability to maintain homeostasis. If allostasis is mild enough and short enough, the organism can recover naturally and reinstate a healthy status. However, if the stress is too prolonged or too severe, then we need a therapeutic intervention to return to a healthy state. Otherwise, we have the emergence of disease as a result of allostatic overload. and an inability of the body to cope with the excess stress that it is subjected to. In the case of Glasgow, we can see that there are very different lifestyles and exposure factors between affluent and deprived areas. The more deprived population is subjected to a variety of stressors. They are typically have more sedentary lifestyles, imbalanced diets, as well as higher levels of psychosocial stress. This causes a number of adverse sequelae, including accelerated telomere attrition, increased adiposity, and poor renal function and hyperphosphatemia. That is also in particularly as a result of excessive consumption of red meat. As a result of this, the deprived are often more biologically aged, and this more biologically aged population is characterized by higher levels of inflammation, chronic inflammation, and bad health outcomes. Traditional measures of biological aging, such as telomere length, are useful. However, they only explain a small proportion, only about 10% of the variation in inflammation. So this leaves the question of where does the other 90% come from? And we believe that a big part is played by the exposome. In our lab, we use the kidney as a model for human normative aging, and we use chronic kidney disease as a model for accelerated aging. This is for a number of reasons. To begin with, chronic kidney disease and biological aging have a two-way relationship with each other. Each one can precipitate the other. Also, the kidney is a very highly metabolically active organ. It has one of the highest metabolic rates of any organ. And it is also exposed to all of the toxins that are produced or absorbed by the body, sometimes in concentrated form. That makes it more susceptible to early damage in case of excessive stress. In addition to that, one of the therapies for chronic kidney disease, which is kidney transplantation, is very interesting from our point of view experimentally, because it involves transferring a healthy organ into a morbid milieu of the CKD patient. So this allows us to track the progression of biological aging in the healthy tissue and in the control tissue and compare the two. In order to measure our outcomes, we're obviously looking for biomarkers of aging, and we are following the Baker and Sprott criteria established in 1988. So we're looking for markers that are functionally related to the process of biological aging, and also that are able to predict physiological and functional capacity at a future date in the absence of overt disease, better than chronological age. This is actually a fairly tall order because chronological age is in fact a pretty good predictor of future morbidity and mortality risk. So this has made it difficult to identify good biomarkers of age. In fact, there are many candidates that have been assessed over the years, but only a few have been validated in humans, and none have achieved the status of universally accepted gold standard biomarkers of age so far. In a lab, we have identified as the best predictor of functionality in kidney transplantation CDKN2A, or the protein P16. This is a cell cycle inhibitor. I'm sure many of you will be quite familiar with it because it's involved in stopping cell cycle, establishing and maintaining cellular senescence. It is also one of the primary ways to detect senescent cells and also to target them therapeutically for clearance in xenotherapeutic interventions. So in our studies, we have seen that CDKN2A is a good indicator of tissue damage of the donor organ. So it is able to predict fairly well whether this organ will function well after transplantation or whether it will fail. our predictive capacity can be further improved if we add additional biomarkers such as certain microRNAs. P16 has also been used successfully in a number of other areas, including vascular aging, as well as to target senescent cells for clearance. And this is one of the key pieces of evidence that has shown that senescent cells are causally linked with biological aging. A more recent biomarker of age that has gained a lot of attention and that we in our lab have moved to is the use of DNA methylation clocks. These are models that estimate biological age or chronological age based on DNA methylation levels at specific genomic loci. We've used three of the more well-established clocks, the Horvath and Hannam. These are perhaps two of the first clocks that gained wide popularity and wide use, as well as the more recent PhenoAge, which is an improvement on the previous generation. The main advance of the second generation clocks is that they are designed and they're trained based on not chronological age like the first generation clocks, but on an estimate of biological age. In the case of PhenoAge, this is a composite biomarker of age that is based on blood values. So the questions that we tried to answer with our pilot study that we run was whether CKD patients indeed display increased epigenetic age. And if this is the case, we wanted to evaluate the two common treatments for CKD, dialysis and transplantation, and assess whether these treatments can ameliorate this accelerated aging. CKD is a progressive disease that goes in stages from 1 to 5, 5 being the most severe case, which is also referred to as end-stage renal disease. And this stage requires therapy, otherwise survival is very low. For this purpose, we recruited patients from a number of different groups. We took samples from CKD stage 5 patients prior to receiving a renal transplantation, as well as patients who were about to start dialysis, as well as age and sex matched population-based controls. We calculated the three clocks as discussed earlier, and we calculated both the epigenetic age as well as the age acceleration, which is the difference between epigenetic and chronological age. We observed that the absolute median error, which is an estimate of how closely the chronological and epigenetic age match each other, is relatively low. It is comparable to previous studies, perhaps only slightly higher. This is reassuring because most clocks are mostly trained on healthy populations, so we couldn't take for granted the fact that they would work accurately in this morbid CKD population. In fact, we also tried a number of other non-epigenetic clocks, particularly a clock based on skin autofluorescence and one based on blood biomarkers, the phenotypic age clock, which was the basis on which the PhenoAge clock was trained, in fact. And both of these non-epigenetic clocks gave us a very high estimate of biological age. Some would argue implausible because they gave us decades higher estimates compared to chronological age, sometimes higher than 100 years of age for patients that were in their 40s, 50s, and 60s. So this was reassuring and confirmed that methylation clocks seem to give a plausible estimate of biological age. They have fairly good correlation with chronological age. If we look closely, we can see that PhenoAge has greater dispersion, so the absolute differences tend to be larger between different samples. What we observed in our cohort was two interesting pieces of information. First, we saw that both CKD5 groups the people about to receive transplant and the people about to go on dialysis, they both showed signs of increased epigenetic age according to all three clocks. So this graph shows number for the pheno-age, which gave us the largest differences and the clearest distinction and the lowest p-values. However, the other two clocks were also consistent with the same trend. So they all showed increased epigenetic age in both CKD5 groups compared to control. And the second interesting finding was that after 1 year after transplantation, the transplant group had partially but not completely reduced epigenetic age. About half of the age acceleration was lost, while we did not observe the same effect in the dialysis group. This led us to conclude that CKD5 patients indeed show signs of accelerated biological aging, at least as measured by epigenetic clocks, and that transplantation partially but not entirely mitigates this age acceleration, unlike dialysis. This is consistent with other findings, both clinical, that see better outcomes and better survival in transplant patients compared to dialysis. In fact, transplantation is considered the superior treatment whenever it is available. And it is also consistent with other studies of different markers, such as telomere length and other genomic DNA methylation patterns that show partial but not complete recovery of the healthy state after transplantation. These are some of the current and recent members of the Shields Lab, as well as some of our collaborators from around the world. I would like to highlight in particular Paul Shields, which is down here, who's the lead of the lab, Colin Selman from the University of Glasgow also, as well as our collaborators from Karolinska Institute, Peter Stenwinkel and his whole team, and Helen Erlandsson in particular, who gathered all the patients and the samples and collaborated with us on this study. And that is all. I will be happy to answer questions, and thank you for the attention.
Great. Thank you very much, Ognian. That's really interesting. And so I think the background of the variations in biological age across Glasgow is something I think is really fascinating. So it's really interesting to hear that background as well. So there's a bunch of questions coming in. I'll start reading them out, and if panelists have a question, then please just jump in or raise your hand. But so the first question is from Ramin Sadri. Does loss of methylation using the DNA methylation clocks reconcile and corroborate cellular senescence and or effects on P16 / CDK N2A? So I guess in other words, then yeah, go ahead.
Do they correlate? So off the top of my head, I cannot think of a study that directly compares head-to-head these two markers in the same patient population. It must have been done, so probably it's just my mistake for not thinking about it. Just a small note, epigenetic methylation clocks don't necessarily measure only loss of methylation. So depending on the locus, some are positively correlated with age, and some are negatively correlated. So it depends on the specific locus. It's not while it is true that overall, generally, the general pattern with aging is to see a general loss of DNA methylation. There are some specific loci that actually do the opposite. They increase their methylation levels with age. So it's a little bit more complicated than that. I would imagine that there should be correlation between methylation clocks and P16, but yeah, off the top of my head, I cannot think of a specific study that directly compares them.
Okay, so I think that Marta Kovacheva had a similar question. Says thanks for the really interesting talk, but again, he's asking about correlations between changes in methylation and P16 levels across the same specific samples. So have you compared methylation and P16 in the same samples?
Not that I recall, again, unless I'm forgetting something very obvious, but certainly not in this study, and I don't think we've. Sorry.
That's okay.
But it will definitely be an interesting thing to look at. That is, that's a very good idea.
Okay. So we have a couple of questions on immunosuppressants, one from Chiara Herzog and another from Ben Parrott. Chiara's asking, is there any indication as to whether immunosuppressant treatment after transplantation might affect epigenetic age independent of the positive renal effects? I'm not quite sure I understand that. And then Ben Parrott wants to know whether immunosuppressants might work through suppressing inflammation as opposed to effects on kidney function.
So these are both excellent questions. We have thought about it. Unfortunately, it's difficult to test because to test that, you would need a control group, which means either you need transplant patients who don't receive immunosuppression, which you can't really do because they need immunosuppression, or alternatively, you would need control patients who receive immunosuppression without disease, without transplant. Again, you can't really do that. So it's a bit difficult to assess that. It's a very interesting question, but we haven't been able to find a suitable control group that would answer that. Interestingly also, one of the possible immunosuppressant that is sometimes used in transplant is rapamycin, which is itself studied as a anti-aging drug. I checked, and as far as I was told, none of our patients have been put on rapamycin. Don't quote me on that, I'm not the expert in this field, but I've been told that my understanding is that rapamycin is more used in North America and less in Europe. So our patients have been put on one or two different immunosuppressants. Whether they have an effect, it's an open question, I guess.
Okay. I mean, I think some of these questions might be, it might help to know a little bit more about the specific causes of the premature biological aging in CKD. You mentioned that kind of in general terms at the beginning.
Yes.
Be more specific about what's known about the causes of accelerated biological aging in CKD.
Yes, of course. So there are a number of factors that to a good extent boil down to the problem of uremia. So when the kidneys don't function well, a number of toxic solutes that the kidney is supposed to be clearing don't get clear. So instead they accumulate in the blood, and that causes different kinds of toxicity. There are a number of molecules that have been identified as playing particularly important role. I'm not sure if we have an exhaustive list of all of the factors that are important. One really crucial one is the role of phosphate. CKD patients have increased serum phosphate levels. This causes premature vascular aging through calcification and increased inflammation. This has all sorts of deleterious effect, because once you have bad cardiovascular health, this sort of precipitates also the health of other organs.
Okay. So if inflammation is a component, I mean, to what extent then do anti-inflammatory interventions, to what extent do they benefit, or is the effect of the phosphate, is it toxic independent of an effect through inflammation?
We believe that it is also independent. It has a role independent of inflammation, because it is it causes calcification. It's a somewhat complex process, but simplified a little bit, the excess phosphate, it leads to formation of calcium protein particles, so crystals of calcium and phosphate bound by proteins. These particles are taken up in the vasculature, especially by smooth muscle cells. They cause damage to these cells. They cause them to convert their phenotype to a more osteogenic phenotype, and therefore they start depositing calcium phosphate in the matrix of the vasculature, and therefore cause all sorts of problems to the vasculature.
Right. Okay. So we have a couple of questions regarding the age of the donor organ. Okay, so to what extent is the effect on epigenetic age dependent upon the donor organ? So would a younger donor organ have a more beneficial effect than an older donor organ, for example?
Yes, I see. So we didn't look at this in the study. I'm not sure even if we have the information about the age of the donors. It is possible. I'm not sure if we have looked at that. It could be interesting. We certainly didn't have the sample size to also look at that. In our case, I would imagine that given functionality of the organ and also biomarkers of age would be perhaps better predictors than pure age, because you can be very young, but if your kidney is not working very well, if you have low filtration rate, EGFR, then it's not going to be as helpful. You're probably better off an organ that is perhaps slightly older but has better functionality. However, it is also true that biomarkers of age and damage, for example, P16 does accumulate with age, so there would be a good correlation between biological and age and function of the organ. So I imagine, yes, it might play a role.
Okay. Margarita Mir has an interesting question regarding the difference between transplantation and dialysis. So she wants to know whether you think that, I guess, the lack of benefit from dialysis, is that due to the fact that dialysis is unable to filter out, some of the detrimental factors within the blood, or is it that dialysis itself is actually a stressful process which may take away some of the benefits?
This is a very good observation, and I think both are likely to be true. Because on one hand, yes, dialysis is not as effective as transplantation in reestablishing normal values of blood biomarkers. At the same time, yes, it is also true that dialysis is a stressful biologically process, so it can contribute. I think in recent years there have been some advances and improvement in different materials and different tweaks to the methodology of how dialysis is performed to reduce this stress. It is a stressful procedure, so that likely contributes as well.
Okay. But independent of that, I mean, do you have a hypothesis as to why transplantation is beneficial, whereas dialysis is not?
I'm not sure what would be the proportion between these two factors. Our hypothesis is that it is due to the fact that the transplanted kidney does a better job at clearing the toxic solutes and therefore reestablishing, fixing the underlying uremia.
Okay. I guess that's something that you could measure, yeah.
Perhaps
you could measure phosphate levels for phosphate levels, for example, after transplant.
Yes. So I suppose we can look at the biomarkers themselves, regardless of, so combine both transplant and dialysis patients, look only at the biomarkers and see how that correlates with epigenetic age. Perhaps, that could be an idea.
Okay.
To look at in the future.
Okay. Paul's has his hand up for a while, I think. Go ahead, Paul. Sorry, Gary. Sorry, I'm sorry. I'm confusing. Gary.
Okay. Ognian, I do have a question. You mentioned earlier socioeconomic differences, and then you mentioned toxins like phosphates. So have you looked at diet and at the microbiome to see if there are impacts there that could also be where you could intervene?
Yes, definitely. This is one of our other areas of research in recent years, because we think that diet plays a major role in health and in outcomes, and the microbiome is increasingly recognized as playing an important role through a variety of mechanisms. It seems to be involved in inflammation and modulation of the immune system, integrity of the intestinal barrier, and leakage of bacteria and toxins through the gut lining is also an important factor. Also, another crucial aspect is the creation of metabolites. So the gut microbiome has orders of magnitude more genes than the human genome, and therefore has a much expanded enzymatic activity and capacity that can create metabolites that our body cannot create by itself, and therefore these metabolites can feed and be absorbed into the body and cause either beneficial or detrimental effects. So yes, this is definitely one of our areas of research. Yeah, we have some recent publication on that line. I didn't address it myself because I haven't been involved too much into this area, so I wasn't really prepared to discuss it in great detail.
Is there a link with is there an epigenetic link?
Between microbiome and epigenetics?
Yeah, well, in this particular case, yeah.
So I'm not sure if we looked directly, so it is plausible because one of the, we believe that one of the potentially important factors is the provision of methyl donors from the diet. So this would be beta-choline that can replenish the S-adenyl-methionine and therefore ensure an adequate supply of methyl donors to maintain DNA methylation. I'm not sure if we have looked directly, specifically measured epigenetic age and microbiome in the same population yet.
Okay, thanks.
But yeah, that would be also interesting, definitely, to see.
So Prasan Datta has a question about infection-induced chronic kidney disease. Is there any evidence that infection-induced CKD causes accelerated aging? And I guess that this is kind of generally interesting because there has been, recently there's been some discussion about the extent to which, acute but resolvable infections can cause accelerated epigenetic aging, and then on resolution, does the epigenetic age actually go back again? Which to me seems like, I know that kind of thing is something that makes me think, well, when we measure epigenetic, when we measure supposed biological age with some of these clocks, I mean, are we really measuring biological age, or are we just measuring something that, for example, reflects acute inflammation? Okay, if it's really going forwards and backwards like that. So what do you know about infection-induced CKD?
Yeah, well, this is one of the big open question, I guess. Yeah, we don't really know what we're measuring with epigenetic clocks. I'm not sure I know much about infection. So this is not acute kidney, it's still chronic kidney disease, but induced by inflammation. I'm not sure. So personally, I don't think I've looked at the different causes that lead to chronic kidney disease and how, if there is any difference between them. So usually when we study CKD, and we don't really look at least in the projects that I've been involved with, we don't really look at the causes what has led to this CKD. I imagine to some extent it shouldn't matter too much because at the end of the day, it's about the functionality of the kidney. So if you have glomerular filtration rate of 60, it is 60. It may or may not depend on what brought you to 60. Of course, it's possible that, yeah, maybe the different causes might cause different type of damage and different type of dysfunction. It's certainly plausible that, yeah, even temporary stress can cause permanent damage to the kidney, because the kidney has a very complex structure that, as I understand, is difficult to regenerate naturally. So typically when you lose units, nephrons, my understanding, so I could be wrong again, I'm not a doctor, but my understanding is that once they're lost, they're pretty much lost. There's very little recovery.
Okay. Well, there's a few more questions in the Q&A that I haven't got to. There's been a lot of questions, so hopefully we'll get those questions forwarded to you and you can answer those.
Yeah, I can try to answer
by email. But I think we need to move on now. So thanks very much, Ognian, for a really fascinating talk.
Thank you.
And